Welcome to this session. This class is especially made for the students of anesthesia. It's only a simple beginning into one of the most fascinating topics we often overlook. Every day in the OT we say things like the patient is down, they are deep, or like we have lost response. But rarely we stop to ask what exactly is going down, what is consciousness, and how do our drugs do it so silently. So this class is just a spark to make you curious. So here are a few questions that we can ask and see for ourselves how many of these can we answer. So the first one is, sleep results from an endogenous rhythm from hypothalamus, brainstem, etc. What exactly is different in anesthesia? Second question, which is the first brain region to shut off during general anesthesia? Which part of the brain creates consciousness, or is there a part at all? Can the cortex be still active while the patient is unconscious? What is the sequence of brain regions shutting off during anesthesia? Or can you tell me the sequence of emergence? Why do some patients have dreams during anesthesia? GABA, NMDA, 2,4-potassium channels, where exactly are all these receptors present? What are the networks associated with consciousness, and which is the network that goes off first during general anesthesia? There are certain definitions given in Miller's textbook of anesthesiology. The first one is regarding consciousness. It is what we lose when we have dreamless sleep and what we regain again in the morning upon awakening. It is a subjective experience. Again, awareness. We inaccurately use the term awareness to include both consciousness and explicit episodic memory. It is a subjective experience. Then comes the terms connected and disconnected consciousness. Connected consciousness is the experience of environmental stimuli, such as surgery, and disconnected consciousness is an endogenous experience, such as a dream state. Now, responsiveness. An individual may fully experience a stimulus, but not be able to respond. So these are certain definitions we have taken from Miller's. We will try and see what these definitions actually mean to us, and in the same chapter we can see the following two images. It shows a part of brain, and there are certain nuclei also mentioned here. We will try and understand which part of the brain is it and what this nuclei actually means to us. So today we are going to have a very superficial look into the gross anatomy of brain, only for the very general details. So what we are seeing here is the grey matter, that is the unmyelinated neurons, and you can see both the left and the right cerebral hemispheres. The structure in the midline is a longitudinal fissure. The part of dura mater that is present inside here is called falx cerebri. You can see certain bumps and depressions all over this structure. The bumps are called gyri and the depressions are called sulcus or sulci. We will learn about other sulci as we move on further. So now we will try to remove certain parts of different lobes of the brain slowly and try to identify what lies beneath. You can see I am removing starting from the right temporal lobe, then the right frontal lobe, then parietal, and finally occipital. So you can clearly see the cerebellum down there along with the entire brainstem. Now that we have fully removed both the cerebral hemispheres, so we are left with this structure. This is the brainstem. Yeah. So that is the brainstem being exposed. You can see the cerebellum also. That's it. And this is the cerebellum being moved away. Now you can have a closer look. This is the brainstem. This is the right putamen, globus pallidus and internal capsule, and the caudate nucleus being removed. And we will be doing similar things on the left side too. Now you can see parts of midbrain, pons and medulla. And you can also see thalamus, hypothalamus, ventricles, the pituitary gland and pineal body. So this is the anterior commissure being taken out, and now you are seeing the blue colored structure that is hypothalamus. isolating the hypothalamus to give you a closer look. That's it. Now coming to the lateral ventricle, choroid plexus. Coming to a closer look on the choroid plexus. A very important structure, isolating the left hippocampus. Coming to the thalamus. So thalamus is very important for us as anesthesiologists. So this is the third ventricle, isolating the third ventricle, taking a closer look. Now you can see we are removing the peduncle of midbrain and then the optic tract. Now we can clearly see the hypothalamus, infundibulum and the pituitary gland. So removing the left lateral and medial geniculate bodies, the midbrain, superior and inferior colliculus. Now the left and right mammillary bodies. Another important structure for us in anesthesiology. These are the left and right mammillary bodies. So coming to the fourth ventricle, this is how the fourth ventricle looks like. So now we have seen the lateral, the third and the fourth ventricle. You can see the pons and the medulla. So we will stop here for the time being. Having said that, now we are coming to the different cross-sectional views of the brain. This is meant to help you out during the ICU rotations where you deal with brain images day in and out. So it is important to figure out which cross-section is being displayed on the CT scan, and thus we can identify the structures involved. Here I will only try and help you to identify various sections, and later on we can try to learn different structures seen on different sections. The first one here is the coronal section of brain. It is also called the frontal section. They provide a view of the brain structures in a front to back orientation. So as you can see, this is how the coronal section looks like. Now coming to the horizontal section. This cuts the brain parallel to the ground, dividing it into superior and inferior portions. This is also known as axial section. So this is how the horizontal section looks like. Now the sagittal section. Sagittal section provides a view of the brain's interior, dividing it into left and right halves. This section reveals key structures like the cerebrum, cerebellum, brainstem, along with the ventricles and other internal components. Okay, now we will quickly see different parts of the brain. So brain can be divided into three parts: the forebrain, midbrain and hindbrain. That is prosencephalon, mesencephalon and rhombencephalon. The forebrain has two parts, the telencephalon and the diencephalon. Telencephalon is mainly made up of cerebral hemisphere and the interventricular foramen. The cavity here is lateral ventricle. The diencephalon contains thalamus, hypothalamus, metathalamus, epithalamus and subthalamus. The cavity here is the third ventricle. The midbrain or mesencephalon contains the crus cerebri, substantia nigra, tegmentum, tectum, and the cavity here is cerebral aqueduct. And hindbrain contains metencephalon and myelencephalon. Metencephalon is made up of pons and cerebellum. Myelencephalon or medulla oblongata is made up of the fourth ventricle. Right? Moving on. So these are the various views of the brain. So this is the First one is the superior view, and here you can see the anterior portion, this is the posterior portion, and these are the left and right lateral portions. The second one is a ventral view. Here you can see the brainstem. Okay, so this is the brainstem, and this is the anterior and posterior view. The third one is the medial view. Okay, so here this is the anterior portion and this is the posterior portion. The fourth one, that is D, is a superolateral view. The fifth one is an anterior view. This is the frontal lobe. These are the frontal lobes. These are the temporal lobes. This you can see the cerebellum, and this is the brainstem. And last one is a posterior view. Yes. So these are the various sections of brain. This is how a section in the coronal plane appears. Like this is a coronal section of the brain. These are the coronal sections. This is how a horizontal section appears. This is how a sagittal section of the brain appears. And an oblique section appears like this. So now you can easily tell which section is this. This is a sagittal section of the brain. You can see different parts of the brain in sagittal section, starting from the cerebrum. This is the lateral ventricle. This is the diencephalon, that is thalamencephalon. Here you can see the thalamus and hypothalamus are situated here. Okay. This is the midbrain, pons and medulla, which makes the brainstem. Here we can see the pons and cerebellum, which makes the metencephalon, and medulla, that's the part of myelencephalon. You can see the cerebral aqueduct here. The midbrain is situated here, and this is the fourth ventricle. This part is the cerebellum. Now this is how the brainstem looks like. This is the midbrain, this is pons and medulla. This is the floor of fourth ventricle. So here you can see how various cranial nerves arise from different portions of the brainstem. This is a sagittal section of midbrain with pons. You can see the pineal body, superior colliculus and inferior colliculus here. The pons is here, and this is a cerebellum. These areas are important for an anesthesiologist to understand how various anesthetic drugs acts on our brain. And here you can see the mammillary body, which is another important part for us. Okay, this is a section of the midbrain where you can see the tectum, tegmentum and crus cerebri. Thalamus and hypothalamus in sagittal section, that is A. Here you can see the thalamus, hypothalamus, the pineal body, cerebral aqueduct, the mammillary body, and B shows superior view of thalamus, and the third one, C, is an MRI of thalamus. Coming to the various parts of thalamus, here is how the thalamus, this is the location of thalamus inside the brain. We will now see various nuclei inside the thalamus. This is the anterior nuclei. Number 2 is the medial nuclei. Number 3A, B and C are lateral nuclei. Fourth one is ventral anterior. Fifth one is ventral lateral nuclei. Sixth one is ventral posterolateral nuclei, VPL. 7 is ventral posteromedial nuclei, VPM. 8 and 9 are lateral and medial geniculate bodies. Again the same portions. Now coming to the various nuclei inside thalamus. The various nuclei inside thalamus are the anterior nucleus, medial nucleus, lateral nuclei, ventral nuclei, intralaminar, midline and reticular nuclei, centromedian nuclei, medial and lateral geniculate bodies. The anterior nucleus is a relay station for hippocampal impulses. It controls emotions and recent memory. Medial nucleus is an area which relates to olfactory impulses and emotions. Lateral nuclei are mainly concerned with correlation. The ventral nuclei is involved in activity of the motor cortex. Ventral lateral nuclei is a relay station for cerebellar impulses. Okay, and coming to the medial and lateral geniculate body. Medial geniculate body is a relay station for auditory impulses and perception of painful and nociceptive stimuli, and lateral geniculate body is for visual impulses. And this is a horizontal section through corpus striatum. We can see the caudate nucleus, the thalamus, globus pallidus, putamen, external capsule, and claustrum. Let's have an overview of consciousness. Consciousness requires active communication between ascending arousal system, thalamus, and cortex, especially the frontoparietal network. Anesthesia disrupts all these three to induce unconsciousness. Now, what is the ascending arousal system or ascending arousal network? It is a subcortical neural network that is critical to consciousness. Ascending arousal network neurons connect the brainstem to the thalamus, hypothalamus, basal forebrain, and cortex, thereby activating cortically based awareness networks. It is a network of interconnected brain structures responsible for regulating wakefulness, alertness, and consciousness. Now we will see which are the various nuclei or regions involved in the ascending arousal system. So coming to midbrain and pons, which is a part of ascending arousal system, there are four main parts: midbrain and pons, hypothalamus, basal forebrain, and thalamus. So in midbrain and pons, the first nuclei is the pedunculopontine tegmental nucleus, which is located in the midbrain. It is involved in wakefulness, REM sleep, and motor control. It contains cholinergic, glutamatergic, and GABAergic neurons. The second one is laterodorsal tegmental nucleus, LDT. It is found near the pedunculopontine tegmental nucleus. It helps regulate REM sleep and wakefulness. Coming to locus ceruleus, it is a small nuclei in the brainstem located in the pons. It is the primary source of norepinephrine in brain. It has a crucial role in regulating arousal, attention, stress response, wakefulness, etc. Its dysfunction can lead to diseases like Alzheimer's, depression, and so on. The raphe nuclei are a cluster of nuclei located along the midline of brainstem. It extends from medulla oblongata to the midbrain. It regulates sleep-wake cycles and is involved in pain perception, emotional responses, etc. It is divided into several regions, including the dorsal median raphe nucleus and raphe magnus. The dorsal raphe nucleus is involved in mood regulation, median raphe nucleus is involved in cognitive functions, and the raphe magnus is involved in pain modulation and motor control. Coming to the ventral tegmental area, the ventral tegmental area of VTI is located in the brainstem. It's located in the midbrain. The role is in reward motivation and addiction. It is a major source of dopaminergic neurons. They project to the nucleus accumbens, prefrontal cortex, and amygdala. The functions of ventral tegmental area are reward processing, dopamine release, emotional regulation, etc. And its dysfunction can lead to psychiatric disorders like schizophrenia, and can also cause major depression. Parabrachial nucleus, it is located in pons. It is also divided into medial, lateral, and subparabrachial nucleus. The medial parabrachial nucleus is involved in taste processing, lateral nucleus is involved in autonomic control, and subparabrachial nucleus is involved in breathing regulation. Therefore, the dysfunction may lead to respiratory abnormalities. Coming to the hypothalamus, the orexin nucleus. It is also known as hypocretin system. It is located in the lateral hypothalamus and plays a crucial role in wakefulness, appetite regulation, energy balance, and reward processing. Orexin neurons project widely across the brain, influencing the locus ceruleus and raphe nuclei. Also, the ventral tegmental area. Dysfunction in the orexin system has been linked to narcolepsy, obesity, depression, and addiction. The tuberomammillary nucleus is a histaminergic nucleus located in the posterior hypothalamus. It has a crucial role in arousal, wakefulness, learning, memory, and energy balance. The tuberomammillary nucleus sends dense projections to various brain regions, including the cerebral cortex, hippocampus, nucleus accumbens, amygdala, and hypothalamus. These connections help regulate cortical activation and behavioral states. Coming to the basal forebrain, it is a region located in the forebrain, situated in front and below the striatum. It plays a crucial role in cognition, wakefulness, attention and memory, primarily through its cholinergic system, which produces and distributes acetylcholine throughout the brain. Nucleus basalis of Maynard is a major cholinergic output to the cortex. The medial septum is a key structure in basal forebrain, playing a crucial role in memory, learning and oscillatory brain activity. It is involved in generating theta rhythms in the hippocampus, which is essential for cognitive functions like spatial navigation, memory consolidation, etc. Coming to the thalamus, thalamus has two kinds of nuclei, the relay nuclei as well as the reticular nuclei. Relay nuclei are specialized thalamic nuclei that act as information hubs, transmitting sensory and motor signals to the cerebral cortex. Examples of relay nuclei include sensory relay nuclei like lateral geniculate nucleus, medial geniculate nucleus, ventral posterior nucleus, etc. Similarly, it has motor and limbic relay nuclei also. So, the lateral geniculate nucleus process visual information, medial geniculate nucleus process auditory information, ventral posterior nucleus (VPN) process somatosensory signals like touch, pain, temperature, etc. Coming to the reticular nucleus, it is located in thalamus as well as brainstem. It is a unique structure that modulates and filters information passing through the thalamus. It plays a role in attention, sleep regulation, and sensory gating. It helps regulate thalamocortical communication, influencing consciousness and sleep-wake cycles. So now we will see what happens during anesthesia. We will have a quick overlook. We will define it in 5 simple steps so that we can have an idea of what happens in our brain in each areas and the nuclei with respect to the action of drugs on different parts of these systems. Coming to step 1, that is suppression of brainstem arousal nuclei. What happens in pedunculopontine nucleus and laterodorsal tegmental nucleus? These are the major neurotransmitter in these nuclei is acetylcholine. So, when anesthetic drugs acts upon these nuclei, it leads to loss of cholinergic tone and cortex becomes less excitable. Locus ceruleus, the major neurotransmitter here is norepinephrine. And what anesthesia does to this region is, it is affected by drugs like propofol, dexmed, etc. and it causes loss of vigilance. Raphe nucleus, which is a major source of 5-HT, and ventral tegmental area, which is a major source of dopamine, on action by anesthetics leads to suppression of mood and motivation. Coming to the parabrachial nucleus, glutamate is a major neurotransmitter. Action of anesthesia can lead to weak relay to the basal forebrain. Yes, step 2 is action of anesthetics on hypothalamus. We have already seen it has 2 nuclei, that is the orexin neurons and the tuberomammary nucleus. In case of orexin neurons, the action by anesthetic drugs can lead to loss of arousal stability. It can mimic a condition like narcolepsy. Tuberomammary nucleus, the major neurotransmitter here is histamine. Action by GABAergic drugs can lead to sedation due to loss of histamine tone. Coming to step 3, the action of anesthetics on basal forebrain. 2 major areas that we have learned in basal forebrain is nucleus basalis of Maynard and medial septum. Nucleus basalis, the major neurotransmitter is acetylcholine, and medial septum, it is acetylcholine plus GABA. So, action of anesthetics on nucleus basalis can lead to decreased cortical arousal as well as attention, and medial septum, that is acetylcholine plus GABA, action of anesthetics can lead to blocking of the hippocampus, leading to decreased signal in the cortical network and blocked memory. Step 4 is the closure of thalamic gate. Thalamus is hyperpolarized by GABAergic drugs. That leads to switching from tonic state to the burst mode. That leads to disconnection of sensory relay, and the reticular thalamic nuclei becomes inhibitory. That will lead to isolation of the cortex. Step 5, it is a cortical network disruption or cortical network fragmentation. Cortical network fragmentation refers to the breakdown or disruption of functional connectivity between different regions of the cerebral cortex. This phenomenon is often observed in conditions such as epilepsy, aging, neurodegenerative diseases, and traumatic brain injury. So, we will now see which are the major networks that are being shut down upon action by anesthetics. Namely, they are the frontoparietal loop, default mode network, salience and executive network, and the EEG. The frontoparietal loops refers to the interconnected neural circuits between the prefrontal cortex and parietal cortex, which play a crucial role in cognitive control, attention, working memory, and decision making. These loops are part of the frontoparietal network, also known as central executive network. So, action by anesthetics upon these loops can lead to loss of connectivity. Coming to the default mode network. It is a large-scale brain network primarily involved in self-referential thinking, memory retrieval, and mind wandering. This network is most active when a person is not focused on external tasks, such as like when a person is doing daydreaming, when he is having some introspection, or recalling past experiences. The key structures of default mode network are medial prefrontal cortex, posterior cingulate cortex and precuneus, and angular gyrus. Okay, so action of anesthetics on the default mode network can lead to loss of self-awareness. Anesthetics will significantly alter the default mode network by disrupting its functional connectivity, leading to a loss of consciousness. The default mode network, which is typically active during wakeful rest and self-referential thinking, undergoes a network fragmentation and the synchronization will reduce under anesthesia. Coming to the salience network and executive control network. These are two major brain networks that play a crucial role in cognitive control, attention, decision making, etc. The salience network is responsible for detecting and filtering important stimuli from the environment and internal states. So it helps switch between different brain networks, particularly the default mode network and executive control network. The executive control network, it is also called the central executive network, is responsible for goal-directed behavior, working memory, and decision making. The salience network acts as a switch determining whether the brain should engage in internally focused cognition, that is action of default mode network, or externally directed task, that is action of the executive network. This dynamic interaction is crucial for attention regulation, emotional processing, and cognitive flexibility. Anesthesia disrupts the salience network and executive control network by weakening connectivity. That leads to impairing attention, fragmenting thalamocortical communication. This breakdown prevents cognitive control and responsiveness, leading to unconsciousness. Finally, coming to the EEG signature. The EEG signature of unconsciousness typically includes increased low frequency power, that is a rise in delta waves. That indicates reduced cortical activity. There is a frontal alpha shift, that is loss of occipital alpha. Occipital alpha is usually 10 to 12 hertz, and that is lost during anesthesia, and there is an emergence of frontal alpha waves which are of lesser frequency compared to occipital alpha waves. So this shift is linked to anesthetic induced unconsciousness. And there is an altered interaction between low frequency and high frequency oscillations which affect sensory processing, and there is network fragmentation like default mode network, salience network, and executive network, etc. Now summarizing, the ascending arousal system suppression will happen first, followed by decoupling of thalamus, and after that fragmentation of the cortex, which in turn and in total leads to a reversible and controlled unconsciousness. So we will have a quick recap what happens during anesthesia. Let's see midbrain and pons. Midbrain and pons, the action of anesthetic can be considered as switching off the engine. So The nucleus involved is pedunculopontine tegmental nucleus and laterodorsal tegmental nucleus. Neurotransmitter is acetylcholine. Effect of anesthesia is decreased cholinergic tone. Cortex becomes less excitable. That will lead to lack of integration. Major anesthetic drugs involved are volatile agents and propofol. In locus ceruleus, the major neurotransmitter is norepinephrine. Effect of anesthesia is diminished cortical alertness. Example, dexmedetomidine propofol. Dorsal raphe nucleus 5-HT is the major neurotransmitter. Reduced serotonin tone will lead to decreased mood and sensory input. In ventral tegmental area, dopamine is a major neurotransmitter. It leads to decrease in the effort-based processes, reward and motivational salience. In case of parabrachial nucleus, glutamate is a major neurotransmitter. Basal forebrain suppression happens mainly by GABAergic anesthetic drugs. In case of hypothalamus, the action of anesthetic leads to desynchronization of sleep-wake cycle. The nucleus involved are orexin nucleus and tuberomammary nucleus. Neurotransmitters are orexin A and B and histamine. Effect of anesthesia causes instability of arousal resembling narcolepsy, especially by propofol, barbiturates and inhalationals. And in case of tuberomammary nucleus, there is fall in histamine tone to the cortex leading to sedation. Example, GABAergic drugs. Action of anesthetics on basal forebrain is like shutting down the attention-related areas. Nuclei involved are nucleus basalis of Maynard and medial septum. Neurotransmitters are acetylcholine and GABA. There is loss of signal in the cortex and decreased modulation of hippocampus leading to memory encoding being affected. That is anterograde amnesia. Action of anesthetics on thalamus leads to sensory relay decoupling. Nuclei involved are intralaminar nuclei. Effect of anesthesia is hyperpolarized by GABAergic agents. They fire in burst mode, and instead of tonic mode, these nuclei will fire in burst mode. That will lead to absence of sustained relay from thalamus to the cortex. Reticulothalamic nuclei becomes hyperactive. There is increased GABA output that leads to shutdown of the thalamocortical loops. Pulvinar or mediodorsal nucleus, there is decreased synchronization between prefrontal and parietal areas, and that can lead to breakdown of awareness. Finally, the cortical networks, cortex and networks, there is fragmentation of workspace. Okay, so there is a theory called fragmentation of workspace hub. There are two main hubs which are involved in that. They are the thalamocortical hub and the corticocortical hub. So, the thalamocortical hub is a central relay system in the brain that integrates sensory, motor and cognitive functions. The thalamus connects with the cerebral cortex through cortico-thalamocortical loops, facilitating information processing and consciousness. It regulates sensory input, attention and executive function by interacting with networks like default mode network, salience network, etc. The key functions of this hub is sensory relay, like visual, auditory and somatosensory signals to the cortex, higher order processing like attention, cognition, memory, and cortical modulation like regulating cortical excitability. The second one, that is the corticocortical hub, refers to the interconnected regions of the cerebral cortex that facilitate direct communication between different brain areas, supporting higher order cognition, sensory integration and executive function. Key features are the frontoparietal network that supports attention, decision making and working memory. On action by anesthetics, this frontoparietal connectivity will collapse. The default mode network, which engages in introspection and memory retrieval. Action of anesthetics will cause shutting down of internal mentation, self-modeling, etc. The salience and executive network, which regulates goal-directed behavior. Action of anesthetics will cause reduced switching and attentional prioritization. It also includes the sensory integration, like visual, auditory and somatosensory processing, which is also affected by action of anesthetics. And EEG will show slow delta and alpha oscillations, which means that cortex is out of sync. Now we will quickly see few drugs and their targets of action. Okay, so ketamine has multiple mechanisms of action. First one, being an NMDA antagonist, it acts mainly in the cortex and thalamus. It decreases glutaminergic excitatory transmission. It also causes thalamocortical disruption. Therefore, the sensory information cannot reach the cortex from the thalamus. Third one is cortex is dissociated from the limbic system. That will lead to distorted perception and blunting of emotional response. Now, fourth one is default mode network disruption. That will lead to loss of self-perception and sense of reality will start to fade. And opioid and monoaminergic pathway contributes to analgesia and euphoria. So these are certain components of the dissociative anesthesia associated with ketamine. So quickly seeing other drugs, propofol, increase in GABA and decrease in glutamate activity leads to suppression of cortex and thalamus, causes EEG anteriorization. Dexmedetomidine, which is an alpha-2 agonist, which will mimic NREM sleep. It will inhibit the locus coeruleus also. Volatile agents, GABA A is increased and potassium leak is increased. There is from 2-pore potassium channels, which will lead to increased global GABA tone and widespread cortical and thalamic suppression. Summarizing again, so anesthesia can be considered as a layered shutdown. Initially, the ascending arousal system is suppressed. That will lead to lack of impulse reaching the cortex or thalamus. Second one, the thalamic hyperpolarization. That will lead to lack of a relay of the impulses to the cortex. And finally, decoupling of cortex. That will lead to lack of integration. So, all these will lead to a combined effect of fade in consciousness, which is a deep and disconnected state when compared to the normal sleep. Now we will quickly see which are the major receptors located in brain and their anesthetic relevance. Coming to the first one, that is GABA A receptors, which are inhibitory chloride channels. They are widespread across cortex, especially frontal and parietal lobes, thalamus, relay and reticular nuclei, brainstem nuclei, locus coeruleus, raphe nucleus and tuberomammillary nucleus, cerebellum and basal ganglia. Major sources of inhibitory tone, they cause sedation and unconsciousness. They are targeted by propofol, volatile agents and benzodiazepines. Coming to the NMDA receptors. They are excitatory glutamate receptors. They are present highly in cortex, especially prefrontal and sensory areas. Hippocampus, they are mainly involved in memory encoding. Thalamus, spinal cord, that is dorsal horn, they are essential for synaptic plasticity and consciousness integration. They are blocked by ketamine. They can cause dissociation and amnesia. The next one is 5-HT or serotonin receptors. The origin is in raphe nuclei. They have widespread projections to cortex, thalamus, hippocampus, basal forebrain and hypothalamus. They modulate arousal, sensory input and mood. They can also be used for analgesia or emesis control. Alpha-2 adrenergic receptors are inhibitory autoreceptors. They are located highly in locus coeruleus, brainstem and hypothalamus. They promote sedation by suppressing the norepinephrine. Dexmedetomidine is an example. It can cause NREM-like sedation. Coming to the histamine or H1 receptors, they are found in cortex, especially frontal lobe, thalamus, brainstem arousal centers. They promote alertness. They are suppressed by GABAergic agents and they can cause sedation. The orexin or hypocretin receptors, the orexin is produced in lateral hypothalamus and have projections to locus coeruleus, tuberomammillary nucleus, PPT, basal forebrain and cortex. They are master stabilizers of wakefulness and are indirectly suppressed by anesthesia via cortical and hypothalamic inhibition. Coming to the 2-pore domain potassium channels, they are found in cortex, hypothalamus, brainstem arousal nuclei. They hyperpolarize neurons and decreases excitability. They are directly activated by volatile agents like isoflurane and sevoflurane. They silence the neurons. So as we have mentioned earlier, we are coming back to the images. So these images are of rodent brain sections. Here you can see certain light green as well as certain dark green colored areas. The light green colored areas are monoaminergic neurons. Monoaminergic neurons are present in the rostral brainstem and caudal hypothalamus. They also innervate cortex as well as subcortical regions. So the monoaminergic neurons will include noradrenergic neurons like in locus coeruleus, serotonergic neurons like dorsal and median raphe nucleus, dopaminergic neurons, that is ventral tegmental area, and histaminergic neurons like TMN. So These are the areas that are involved in the neurobiology of wakefulness. There are also certain other wake promoting areas which include the cholinergic regions, which are the dark green colored areas with hatching, like pedunculopontine and lateral-dorsal tegmental areas, and basal forebrain. General anesthetics have been demonstrated to suppress many of these regions. Coming to the second image, we can see GABAergic neurons in the ventrolateral preoptic area and median preoptic nucleus, the hypothalamus. These are sleep promoting areas. So in this image, you can see the ventrolateral and median preoptic nucleus, that is the lowermost in blue color. These are GABAergic neurons, and these neurons will promote sleep by inhibiting the wake promoting neurons in the caudal hypothalamus and the brainstem, as we have already mentioned. These hypothalamic nuclei are activated by the general anesthetic. So we are wrapping up this session here for the time being. This class is an invitation to think deeper. Anesthesia isn't just about putting the patient down, it's about understanding the very nature of consciousness. I hope this class sparked more questions than answers, because that's where real learning begins. If this session added even a drop of clarity to your knowledge, please let me know and feel free to share your suggestions.